everyone, and welcome to today's webinar titled Unlocking the Potential of Cancer Therapeutic Antibodies, GenScript's Immune Checkpoint and Therapeutic Antibody Solutions. This webinar is a part of the 11th annual event in the Cancer Research and Immuno-Oncology Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, please go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You will also notice that the speaker has provided a survey located on the right of your screen. Please take a few minutes to answer the survey questions. It will be available during both the live session and on-demand period. And if you're having trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the event. Now let's get started. I'd like to present today's speaker, Amanda Grimm, Senior Segment Marketing Manager, Antibody Drug Discovery at GenScript USA. For a complete biography on our speaker, please find it on the left of your screen. Amanda, you may now begin your presentation.
Great, thank you so much, Kaylee, for the introduction. Let's go ahead and get started here. As mentioned, my presentation today is titled Unlocking the Potential of Cancer Therapeutic Antibodies, and I'll be providing you with details about GenScript's comprehensive immune checkpoint and therapeutic antibody solutions. Some key considerations about GenScript and their technology in case you are not familiar with us or are thinking about wanting to work with us after this presentation. We have over 20 years of development experience, 18 years of dedicated antibodies service specifically, and we've completed over 150,000 antibody projects to date. Some history and milestones about the GenScript group. We were founded in 2002 here in New Jersey, USA. Since then, we've added some subsidiaries like our BestSime, Legend Biotech, and our CDMO ProBio. Something to note, in 2017, we joined in a partnership between Legend and Janssen with the Cilta-cel, which then received its FDA approval last year in 2022. So great things coming through our pipeline. The contents of my talk today will include some background and history about antibody therapeutics. I'll go into the various solutions GenScript offers for antibody drug development, and then I'll touch on what it looks like to work with us. So we'll start with antibody therapeutics. So the first slide here, what are antibody therapeutics? Just to add some definition, they're used to treat a disease. The antibody is typically a monoclonal and would be a whole molecule or a fragment. Often the antibodies are modified to improve function. We see a lot of bispecifics in the cancer research and therapeutic development space, and these are important because of the T-cell engager, in order to modulate that immune response. We're also seeing trifunctional antibodies on the increase as well. We're also seeing antibody drug conjugates. These are on the rise, and these are antibodies that include a payload with a linker, depositing that payload into its target. So here we'll look at the antibody therapy history. The first approval was in 1986. After that initial approval, it was a little bit of a slow development, new technology. It isn't until 2006 that we start to see approvals on an annual basis. By the time we get to 2021, This marks the 100th approval by FDA, and this is particularly with the SARS-CoV-2 monoclonal therapy that we should all be pretty familiar with. Here we have a breakdown of the history as far as cancer versus non-cancer. Over half of the therapeutics really are directed towards cancer, as depicted in the red, and the chart itself really indicates approvals in the US or EU each year from 1997 to 2022. Here we have a list of the top 10 approved antibody drugs. Notably, we have cancer. We also have the second highest, which is autoimmune diseases. But representing cancer in this top 10, we see melanoma, small cell lung cancer, colorectal cancer, and breast cancer. And then to look at one of these a little bit further, we'll look at Keytruda. It's an example of a checkpoint inhibitor. It's manufactured by Merck and was approved in 2014. Its target is PD-1. PD-1 is an immune checkpoint receptor that, when bound, turns off the T cell activation. So Keytruda comes in, it blocks PD-1 on the T cell from binding PD-L1 on the tumor cells, and now T cells can be activated and the proper pathway enacted to kill melanoma cells. And the image on the left is a depiction of what this looks like, of the pathway. So let's go ahead and move into GenScript's solutions for antibody drug development. And here I have a general overview of the workflow for therapeutic antibody development. We've essentially broken this down into 4 stages. We have target identification, lead generation and optimization, preclinical, and then clinical development. And then in this slide just kind of takes that a step further, where we're looking at the individual stages and all the different products and services that GenScript can offer as it relates to each stage. We have a lot listed. I won't be going through all of these today, but just know that if there's something that you're having a question about, we can probably help you. So as always, don't ever hesitate to reach out to us to ask us questions. So we'll start with the first phase, which is target identification. Here we understand that you're working diligently to identify the drug target, determine if it's druggable, and understand the associated disease mechanism. This can happen through a variety of methods, whether you're using AI, reading through all the literature, maybe there's already information known to you, maybe you've already started conducting some experiments. Here we've broken it down into our solutions as far as gene analysis, protein analysis, and cell-based analysis. And in that first section, we'll go ahead and start at the gene level. Your research may involve a need to amplify a target using PCR. You can measure the DNA or RNA levels using qPCR or RT-qPCR. GenScript offers ready-to-use reagents, as well as offering our oligo service that creates probes and primers specific to your project. Targets can be further investigated using gene editing in order to simulate the disease condition or to correct it. We have a variety of options for off-the-shelf CRISPR-Cas9 plasmids and enzymes to support that evaluation of target genes and the functionality. If you are also ordering something that's going to incorporate functional guide RNA, this will be generated specific to your project. The next step we can look at with gene level analysis is the Gene Titan 8 million oligos, next-gen sequencing, and CRISPR, gRNA libraries. These allow you to further accelerate your target discovery efforts. These tools enable comprehensive exploration of target genes, precise gene editing, and deep insights into gene function. The Gene Titan 8 million oligo pools provide pools of diverse sequences that can be used for target identification and validation. These high-quality oligos are designed to cover a wide range of target genes that enable the exploration of a broad landscape of potential targets. Our NGS is enabled by Gene Titan. It allows for high-throughput sequencing and analysis of genetic material. Our NGS capabilities enable insights into the target genes, their expression profiles, and potential functional implications. This information can then help to prioritize targets for further downstream investigation. CRISPR gRNA libraries are a cutting-edge technology for efficient gene editing and functional genomics. We offer a comprehensive collection of guide RNA sequences that target specific genes or pathways of interest. Some examples include acute myeloid leukemia, as well as bladder cancer. So within target identification, we can look at protein analysis. And if you're doing in-house analysis, we offer gels and blotting automation as fundamental tools for protein detection and analysis. This includes the blotting units, membrane processing instruments, electrophoresis standards, and high-quality antibodies for screening. Automating Western work is a must, with the amount of time saved from making the gels, avoiding uneven transfer, and 1 million tubes or trays for staining. We also offer a variety of immune checkpoint proteins and antibodies as off the shelf. They're essential for studying the immune system and developing your immunotherapies. Taking a look at enabling antigen creation and protein interaction, GenScript's protein expression service covers multiple expression systems to accommodate the diverse needs and optimize the production of target proteins. Efficient and reliable protein expression is crucial for obtaining sufficient quantities of target proteins for characterization and functional analysis. For the more simple protein expression needs, the E. coli platform provides a cost-effective and rapid solution. It is ideal for the expression of soluble proteins, protein fragments, or small-scale production of antibodies for initial screening and characterization. The insect expression platform offers an alternative for the production of those challenging proteins that require proper folding and post-translational modification. This system utilizes insect cells to produce recombinant proteins with high yield and functionality. Lastly, the mammalian expression platform offers the advantage of post-translational modifications and native folding, making it suitable for the production of the more complex proteins and antibodies that require proper folding and glycosylation. So by the time we have expressed our target, we may be looking at the functionality of that target. And so for target validation, frequently we might have hundreds and even thousands of samples that need to be analyzed. This can be hard to do with Western, so ELISA kits are often used due to easy adaptation to high throughput and automation. By leveraging our expertise in antibody production and assay development, you can obtain highly sensitive and specific ELISA kits tailored to your target protein. The kits provide a robust platform for screening and identifying potential therapeutic targets that's then going to allow you to make informed decisions about your antibody drug development efforts. Taking a look at the cell-based assays, our CRISPR cell lines offer a revolutionary approach to genetic manipulation, allowing for those precise and efficient genome editing. GenScript's CRISPR cell line services enable you to generate stable cell lines with the precise modifications, such as gene knockouts, knock-ins, or point mutations. These cell lines serve as valuable tools for functional validation, target identification, and later lead optimization. Our cell power cell line development services offer a comprehensive solution for optimizing cell lines specifically for antibody production. Our team utilizes advanced cell engineering techniques in order to enhance cell line performance, and that includes increased productivity, improved protein quality, and enhanced stability. Our off-the-shelf stable cell lines are a valuable tool in antibody drug target validation. They're going to provide a consistent and reliable source of cells expressing protein at a known level. That is then going to enable you to evaluate the function and identify potential drug candidates that selectively target the protein. So here we have a summary of the solutions for target identification. Again, that covers gene analysis, protein analysis, and cell-based analysis solutions. After your target is successfully expressed, purified, and confirmed, antibody candidates can now be generated for screening and optimization. And here we've just broken that down essentially into two groups, lead generation and identification, and then of course optimization, outlining the different solutions that we offer. When we look at plasmid design and construction, our goal is to simplify this with your gene and plasmid design. And so we offer our GenSmart intelligent platform. The GenSmart DNA construct design tool is free and easy to use. It's going to expedite the plasmid design process. For example, you can select from two modes, either creating a construct or creating a library if you need batch design. Ultimately, when you're done, you can send for a quote and or place the order directly from the website. We also have the codon optimization feature, which has additionally been upgraded to ZQ. And this means that it now employs a deep learning powered system in order to enhance the ability to express those difficult proteins and results in a significantly higher yield. Next, we have GenScript's gene synthesis service. This provides efficient and accurate synthesis of custom DNA sequences. With our expertise in gene synthesis, we're able to obtain high-quality DNA constructs tailored to the specific antibody targets and optimization needs. This service facilitates the generation of diverse antibody variants, allowing for screening and selection of lead candidates with improved properties such as binding affinity, specificity, and stability. In order to expedite the process of DNA assembly and cloning, our DNA assembly kit offers a powerful tool for seamless and precise assembly of DNA fragments. The kit employs a user-friendly one-step assembly method that allows you to rapidly construct complex DNA libraries or variants for lead optimization. Our catalog products division offers automated plasmid purification solution as their new AMAG Quattro. The Quattro is a modular design capable of purifying up to 24 samples with 6 samples per module, all controlled from one unit. The modules can be purchased individually or as a package, and they offer precise pipetting capabilities with pre-packaged reagent consumables that are placed into the instrument and the process initiated using the easy-to-use interface. You can expect yields of 300 micrograms to 1.5 milligram of plasmid with low endotoxin levels completed in a runtime of approximately 2 hours. Moving on to antibody development. First, we'll look at our mouse hybridoma monoclonal antibody platform. This is the more traditional method, but it is the most common in order to generate monoclonals. We can provide custom antibodies from mouse or rabbit. This workflow includes the option for you to provide us with your antigen sequence, and we'll make the antigen, or you can provide your purified protein for immunization. Our turnaround to have hybridoma antibody from gene to customer is approximately 14 to 23 weeks. We have the options for our standard or custom packages, each with the option to add our proprietary monoboost to enhance the immunization and shorten that timeline. Also to note too, we're very flexible on the immunogen type. It can be protein, peptides, small molecules, DNA, etc. Our other custom antibody development is our monorab, rabbit monoclonal antibodies. The rabbit immune system is known for producing high affinity and highly specific antibodies. Based on this technology, antigen-specific B cells are isolated and enriched, followed by screening to identify key antibodies. Those antibody candidates are then sequenced and recombinant antibody produced. We also have the option to add the Beacon B cell screening platform to this development process. The Beacon is a high throughput instrument that screens a high number of individual B cells, performing multiple screening tests in one place. So what normally may take a week or more can be condensed into one day. As noted in the hybridoma overview, we offer a number of different options for immunogens. Here I wanted to provide a quick intro to our mRNA immunization that was just recently launched. This particular immunization offering is going to help ensure proper expression of those difficult to express proteins like GPCR, membrane protein. We do have some data from a case study we did if you're interested in seeing how we were able to improve lead outcomes with this method. Our TurboMab is also a new service I wanted to touch on here, just to kind of highlight and pinpoint that after immunization, we would do positive cell enrichment before performing the fusion. This is going to ensure that you get the most leads as possible that are also specific. We can also incorporate the Beacon into this process as well in order to expedite that B cell screening and get you your candidates in as soon as 8 weeks. Another instrument that may help in your protein or antibody purification comes from our catalog products division and is our immunoassay Plus. It's a semi-automated instrument providing high throughput purification of up to 12 samples in less than an hour with results in up to 80 milligrams of protein per sample. The AMAG process cuts down multiple steps compared to the traditional methods by adding the magnetic beads directly to the samples, and then the instrument takes care of all the washing and luting for you. Taking a look at optimization, our TurboCho platform is a proprietary protein expression platform developed by GenScript. It includes an option suitable at each stage of development with variables based on protein yield, production times, and the deliverable characteristics. It's a scalable platform that provides at the high throughput level transient expression of 1 to 20 mils. It's our fastest at a 5 business day turnaround from gene to protein or antibody purification. It provides a yield of 0.1 to 10 milligrams. This level is most complementary to earlier target discovery phases. When we look at the express tier, it is also a transient expression, but at a larger scale of 40 mils to 200 liters. The timeline is going to be 10 to 14 days, providing you with yields from milligrams to grams. It's also capable of providing expression of diverse antibody formats, like single domain antibodies, BITEs, and bispecifics across mouse, human, and rabbit. And then we also have our high performance. This is our stable expression scaling up to 200 liters. The timeline is going to be 6 to 7 weeks, providing yields ranging from grams to kilograms. This stage supports this later optimization goal in order to generate those larger, more stable expression volumes that are going to be needed to support preclinical studies and IND filings. So lead optimization in antibody drug development often involves the exploration of diverse antibody variants with improved characteristics. Our mutant libraries offer a comprehensive collection of pre-designed or custom-designed variant libraries that cover a wide range of mutation types. These libraries provide the ability to screen large numbers of antibody variants to identify leads with enhanced binding affinity, specificity, stability, or other desired properties. Our precision mutant libraries are powered by our GeneTitan platform, enabling the improvement of antibody binding affinity via CDR engineering and antibody affinity maturation. This can be done through saturation scanning, site saturation, and or combinatorial mutagenesis in order to create a unique library for your project. This slide outlines the workflow information related to our humanization service that's going to allow you to tailor and improve the characteristics of your antibody candidates for enhanced efficacy and reduced immunogenicity. Our customers provide us with their monoclonal antibody sequence, or we can even assist from the development, starting with those earlier discovery phases around antigen design as well. The humanization process is based on computational algorithms and molecular modeling to modify those non-human antibodies in order to make them more compatible with the human immune system while preserving their target binding characteristics. By minimizing potential immunogenicity risks, humanized antibodies have a higher likelihood of progressing through preclinical and clinical development stages. Our service will generate up to 15 humanized monoclonals. Our scientists will validate using ELISA binding and EC50 affinity ranking, and then the top 5 will be provided to you as a small-scale transfection so that you can do further validation in your hands. So just to summarize the lead generation and optimization, we covered solutions for plasmids, antibodies, functionality, and optimization. So moving on to preclinical, at this point you've confirmed antibody candidates, and you're ready for those preclinical evaluations and studies. In this stage, it's important to perform many tests according to the regulatory criteria to assess and collect data related to that toxicity and PK. Non-human animal studies are incorporated as well to determine whether the antibody performs as intended and whether it's going to be safe. Here, we've just broken this down into candidate validation as well as IND study, and then listed out the different products and solutions. So we'll start here with GenScript's plasmid prep. We do also include research and industrial grade levels. However, here I do want to highlight our preclinical grade that's going to ensure that plasmid production is completed with those higher standards and extensive QC testing. The chart here outlines what you can expect from the service for specific QC parameters and which method is used. Some QC examples to note are sequence verification and DNA purification and confirmation. The goal is to ensure our plasmid prep service delivers your plasmid DNA with high purity, yield, and consistency, and that also guarantees 100% sequence accuracy. GenScript offers valuable tools in order to evaluate the pharmacokinetics, immunogenicity, and functional characterization of your potential antibody therapeutics during the preclinical development. Anti-idiotype antibodies play a crucial role in the development and characterization of antibody drugs. They are designed to specifically recognize and bind to the unique variable region of another antibody, also known as the idiotype. Anti-ID antibodies are generated using advanced recombinant antibody technology, ensuring high specificity and sensitivity. We also have a large collection of catalog anti-idiotype antibodies as well. Within this, we provide 3 types of antibodies, those involved in detecting free antibody through antigen blocking, the second detecting total antibody through non-blocking mechanism, and then the last detects bound antibody that is drug target complex specific. These can be combined with our custom DNA kits and, again, our powerful tools for quantitative analysis for conducting essential PK and ADA assays. Cytokines and growth factors are needed in cell culture media in order to promote proliferation and differentiation. They're especially critical when serum-free media is used. We do want to highlight that we offer a large number of cytokines and growth factors, and that these are produced under ISO 13485 for critical cell culture needs. Our gene to lentivirus service enables you to efficiently generate high-quality lentiviral particles carrying the gene of interest, facilitating the functional analysis and in vitro validation of antibody drug candidates. The service begins by cloning of the gene of interest into the lentiviral vector, which is optimized for efficient gene delivery. The lentiviral particles are then produced through a state-of-the-art lentivirus packaging system, generating high-titer viral stocks. These lentiviral particles can be used to transduce a wide range of cell types, including both dividing and non-dividing cells. By introducing the gene of interest into target cells, you can assess its functional properties, evaluate its biological activity, and study its impact on cellular pathways relevant to your antibody drug development. During the preclinical stage, it's also crucial to have ample supply of antibodies for in vitro and in vivo studies, including functional characterization, efficacy, evaluation, and safety assessment. Our large-scale antibody production service offers a scalable and customizable approach to meet the specific demands of preclinical research, ensuring high purity and biological activity. Whether it's monoclonal antibodies or recombinant antibody fragments, such as single-chain variable fragments or Fab fragments, our production capabilities can accommodate a diverse antibody format. Our services also follow strict quality control measures, offers flexible scalability, competitive prices, and strict confidentiality. GenScript's ProBioBiologics CMC service offers comprehensive support for preclinical and clinical stage of antibody drug development. This service is specifically designed to address the critical aspects of biologics development, including process development, manufacturing, and quality control, ensuring that smooth transition from preclinical research to clinical trials. Our ProBio team offers an expertise in process optimization and scale-up, ensuring the efficient and consistent production of antibody drugs for clinical studies. So here we have just a summary of what we've covered in this preclinical stage. Our GenScript solutions include a range from research use only through the GMP grade products and services, and include services such as the biologics CMC, offering cytokines and growth factors, large-scale antibody production, anti-ID antibodies, and lentivirus engineering. So this is our last stage of solutions related to your clinical development. I've previously mentioned our ProBio team in the preclinical with the biologics CMC, and I do want to emphasize the ease of transition of technical transfer as the quality scale increases, because we do have our CDMO integrated into our processes. These final services that I'll share are provided by our highly experienced CDMO team. In this final stage of clinical experiments, the complexity increases and each stage requires very controlled studies involved. I just wanted to provide this clinical trial overview as a summary of the clinical trial process that I took from the US FDA website. Here we've broken down the offerings into cell banking, manufacture, and then analytical services and products. And the first of these that we'll look at is the generation and characterization of high quality stable cell lines for the production of antibody drugs intended for clinical studies. During the clinical stage, it's crucial to have a well-characterized and scalable cell line that consistently produces the desired antibody with that high yield and quality. The GMP cell line development and cell banking services cover various activities, including cell line selection, genetic stability assessment, productivity, optimization, and establishing GMP compliant master and working cell banks. Our ProBio team follows strict quality control procedures and conducts comprehensive characterization and testing to ensure the safety, stability, and production of those cell lines. During the last stages of antibody drug discovery, it's critical to transition from clinical studies to large-scale manufacturing. ProBio's ProGMP manufacturing service addresses this need by providing that seamless and compliant pathway for the production of antibody drugs for commercial use according to regulatory compliancy and CGMP requirements. ProGMP manufacturers covers a wide range of activities, including the process optimization, scale-up, tech transfer, CGMP manufacturing. ProBio works really close with our clients in order to develop that customized manufacturing strategy to meet the specific requirements of their antibody drug candidates. And here we'll summarize the clinical development as far as providing clinical analytics, as well as, importantly, the GMP development and manufacturing. And our last topic, I'll go into what it looks like to partner with GenScript. So if you're curious about what a partnership would look like, it really starts with a call to learn about your project and gives you the opportunity to ask questions. We ask questions, and then we find out the needs that you may have. The therapeutic antibody development process is complex and likely includes many projects within the scope of what you need. So each partnership is going to be unique and it's going to be customized to our clients' needs. To do that, we ensure that you have a dedicated project manager to your case to be with you throughout the process in order to keep you informed, ensure timelines, and make sure you're getting the technical and ongoing support that you need. And with that, I'll say thank you for your time today. I appreciate your attendance. And as always, we're always here to answer questions. Don't hesitate to reach out and be sure to visit our booth. Thank you.
Wonderful. Well, thank you, Amanda, for your informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question that you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. All right, so let's get started. Our first question here asks, what is the advantage of using the Beacon platform to the regular single-cell workflow?
Yes, great question. The Beacon itself is an instrument, and it allows us to screen thousands of B cells individually and at once. It also has the capability to perform multiple screening tests at the same time. So all of this can be done in one day versus traditional methods that can take weeks.
Great, thank you. The next question we have asks, are you able to offer humanization for other types of antibodies such as mouse or camelid?
Yes, we have capabilities to do this for any species.
Great. Another question here. What is the maximum amount of protein or antibody that can be purified on your automated purification system?
Yes, great question. So the AMAG assay, you would get 80 mg per sample, and that can hold up to 12 samples.
Great. Well, it looks like we have time for one last question, and this question asks, did I understand correctly that with your TurboCHO platform, you can express antibody from gene synthesis to purified antibody in 5 days?
Yes, this is going to be applicable to our high throughput tier. It's our most cost-effective option for small-scale recombinant antibody or protein. We've expanded the service to be high capacity in order to accelerate therapeutic development.
Wonderful. Well, thank you, Amanda. This does conclude our session today. Do you have any final comments for our audience before we go?
I do not. Just thank you so much for your time. And again, check out our booth. We have a number of references there available for everybody to be sure to get.
Well, thank you again, Amanda, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until October 7th, 2025. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.